Translation. By the end of this video, you will be able to describe the different steps in protein synthesis, discuss the role of ribosomes in protein synthesis, and describe the genetic code and how the nucleotide sequence determines the amino acid and the protein sequence. The synthesis of proteins is one of a cell's most energy-consuming metabolic processes. In turn, proteins account for more mass than any other component of living organisms, with the exception of water, and proteins perform a wide variety of the functions of a cell. The process of translation, or protein synthesis, involves decoding an mRNA message into a polypeptide product. Amino acids are covalently strung together in lengths ranging from approximately 50 amino acids to more than 1,000. Welcome back to Concepts of Biology. We'll continue to read Chapter 9, Molecular Biology, Section 9.4. The Protein Synthesis Machinery. In addition to the messenger RNA template, many other molecules contribute to the process of translation. The composition of each component may vary across species. For instance, ribosomes may consist of different numbers of ribosomal RNAs, or rRNAs, and polypeptides depending on the organism. However, the general structures and functions of the protein synthesis machinery are comparable from bacteria to human cells. Translation requires the input of an mRNA template, ribosomes, tRNAs, and various enzymatic factors. Figure 9.19. The protein synthesis machinery includes the large and small subunits of the ribosome, messenger RNA, and transfer RNA. In E. coli, there are 200,000 ribosomes present in every cell at any given time. A ribosome is a complex macromolecule composed of structural and catalytic rRNAs, ribosomal RNAs, and many distinct polypeptides. In eukaryotes, the nucleolus is completely specialized for the synthesis and assembly of ribosomal RNAs. Ribosomes are located in the cytoplasm in prokaryotes and in the cytoplasm and endoplasmic reticulum of eukaryotes. Ribosomes are made up of a large and a small subunit that come together for translation. The small subunit is responsible for binding the messenger RNA template, whereas the large subunit sequentially binds transfer RNAs, a type of RNA molecule that brings amino acids to the growing chain of the polypeptide. Each messenger RNA molecule is simultaneously translated by many ribosomes, all synthesizing protein in the same direction. Depending on the species, 40 to 60 types of transfer RNA exist in the cytoplasm. Serving as adapters, specific transfer RNAs bind to sequences on the messenger RNA template and add the corresponding amino acid to the polypeptide chain. Therefore, transfer RNAs are the molecules that actually translate the language of RNA into the language of proteins. For each transfer RNA to function, it must have its specific amino acid bonded to it. In the process of transfer RNA charging, each transfer RNA molecule is bonded to its correct amino acid. The genetic code. To summarize what we know to this point, the cellular process of transcription generates messenger RNA, a mobile molecular copy of one or more genes, with an alphabet of A, C, G, and uracil, U. Translation of the messenger RNA template converts nucleotide-based genetic information into a protein product. Protein sequences consist of 20 commonly occurring amino acids. Therefore, it can be said that the protein alphabet consists of 20 letters. Each amino acid is defined by a 3-nucleotide sequence called the triplet codon. The relationship between a nucleotide codon and its corresponding amino acid is called the genetic code. All right, let's get back to reading. Given the different numbers of letters in the messenger RNA and protein alphabets, combinations of nucleotides corresponded to single amino acids. Using a 3-nucleotide code means that there are a total of 64, that is 4 times 4 times 4, possible combinations. Therefore, a given amino acid is encoded by more than one nucleotide triplet. Figure 9.20. This figure shows the genetic code for translating each nucleotide triplet or codon in messenger RNA into an amino acid or a termination signal in a nascent protein. 3 of the 64 codons terminate protein synthesis and release the polypeptide from the translation machinery. These triplets are called stop codons. Another codon, AUG, also has a special function. In addition to specifying the amino acid methionine, it also serves as the start codon to initiate translation. The reading frame for translation is set by the AUG start codon near the 5' end of the messenger RNA. The genetic code is universal. With a few exceptions, virtually all species use the same genetic code for protein synthesis, which is powerful evidence that all life on Earth shares a common origin. The mechanism of protein synthesis. Just as with messenger RNA synthesis, protein synthesis can be divided into 3 phases: initiation, elongation, and termination. The process of translation is similar in prokaryotes and eukaryotes. Here we will explore how translation occurs in E. coli, a representative prokaryote, and specify any differences between prokaryotic and eukaryotic translation. Protein synthesis begins with the formation of an initiation complex. In E. coli, or Escherichia coli, this complex involves the small ribosome subunit, the messenger RNA template, 3 initiation factors, and a special initiator transfer RNA. The initiator transfer RNA interacts with the AUG start codon and links to a special form of the amino acid methionine that is typically removed from the polypeptide after translation is complete. In prokaryotes and eukaryotes, the basics of polypeptide elongation are the same, so we will review elongation from the perspective of E. coli. The large ribosomal subunit of E. coli consists of 3 compartments. The A site binds incoming charged transfer RNAs, that's transfer RNAs with their attached specific amino acids. The P site binds charged transfer RNAs carrying amino acids that have formed bonds with the growing polypeptide chain but have not yet dissociated from their corresponding transfer RNA. The E site releases dissociated transfer RNAs so they can be recharged with free amino acids. The ribosome shifts 1 codon at a time, catalyzing each process that occurs in the 3 sites. With each step, a charged transfer RNA enters the complex, the polypeptide becomes 1 amino acid longer, and an uncharged transfer RNA departs. The energy for each bond between amino acids is derived from GTP, a molecule similar to ATP. Amazingly, the E. coli translation apparatus takes only 0.05 seconds to add each amino acid, meaning that a 200 amino acid polypeptide could be translated in just 10 seconds. Figure 9.21. Translation begins when a transfer RNA anticodon recognizes a codon on the messenger RNA. The large ribosomal subunit joins the small subunit, and a second transfer RNA is recruited. As the messenger RNA moves relative to the ribosome, the polypeptide chain is formed. Entry of a release factor into the A site terminates translation, and the components dissociate. Termination of translation occurs when a stop codon, for example UAA, UAG, or UGA, is encountered. When the ribosome encounters the stop codon, the growing polypeptide is released and the ribosome subunits dissociate and leave the messenger RNA. After many ribosomes have completed translation, the messenger RNA is degraded so the nucleotides can be reused in another transcription reaction. In our next video, we'll read Section 9.5, How Genes Are Regulated.